Good morning, all of you. In last practical, I have discussed to perform the microbiological assay of crystalline sodium salt and to study to study or to perform the sterility test of sodium chloride injection. Today, we will see the next practical for syllabus, that is to carry out the bacteriological analysis of water. So, aim of today's practical, that is to carry out bacteriological analysis of water. First, we will see introduction about this practical. So, water is the major source of different causative agent of gastrointestinal diseases. Therefore, it is must be essential to maintain the purity of water. What is need of or what is necessity of maintaining the purity of water? Because as it is the major source of different causative agent of gastrointestinal diseases. Then, when water is used for human activity, especially for drinking water, it should be highly purified. The different type of microbes such as fungi, protozoa, algae, then actinomycetes, bacteria and viruses are found in water. There are different types of microbes which are found in water. These microbes are causative agents of various diseases like dysentery, that is Entamoeba histolytica, then cholera, that is Vibrio cholerae, then typhoid fever for Salmonella typhi. Then the purity of drinking water is evaluated by testing the presence of coliform, that is indicator microorganism, as evidence of fecal contamination. For example, E. coli, that is Escherichia coli, then Streptococcus faecalis, then Clostridium welchi. The E. coli is the most predominant coliform present in the intestine of human and other animals. Then the water is considered safe for drinking only if it is not less than 4 fecal coliform that per 100 ml, as determined by the membrane filtration method or the membrane filter method. Then coliform bacteria in water are also detected by a multiple tube fermentation test, then immune test and the total plate count technique. Then we'll see the sample media. For that, water samples from sewage plant or the ponds or the tap water. Then media, double and single strength lactose fermentation broth, then nutrient agar medium, then eosine methylene blue medium, and last, that is McConkey's agar medium. Then reagents used for this practical, that is crystal violet, grams iodine, ethyl alcohol and safranin. Then apparatus for this pipette, fermentation tube, then glass marking pencil, conical flask, nichrome wire loop, Durham tubes, slides and gram stain reagents. The equipment required for this incubator and the autoclave. Then we will see the principle of this practical. The coliform group include all aerobic, then facultative, anaerobic, then gram negative, then non-spore forming, then the rod-shaped bacteria that ferment the lactose with gas and the acid formation within the 48 hours at 70 degrees Celsius. Then in multiple tube fermentation method, the property of bacteria to ferment the nutrient broth like lactose to produce a gas and the acid within the 48 hours at a temperature, that is 37 degrees Celsius, is utilized for detecting the presence coliform bacteria. Then subsequently, the presence of fecal coliform is differentiated when the bacteria on fermenting lactose produce a gas in a relatively shorter period of time and at a relative higher temperature, that is 24 at 44 degrees Celsius. Then this method requires testing of 3 sets of dilutions. Then the standard test for coliform group may be carried out either by the multiple tube fermentation technique through the presumptive and the confirmed phase. or by the membrane filtration technique. The standard test for this coliform group, it may be carried out either by the multiple tube fermentation technique or the membrane filtration technique or the membrane filter technique, that is capital MF. Then the coliform bacteria in water are detected by three tests. First, that is presumptive test, then confirmed test, and third, that is completed test. This test detects the presence of coliform bacteria, that is indicator of fecal contamination. The gram-negative, then non-spore forming bacilli that ferment the lactose, that is carbon source, with the production of what? That is production of the lactic acid, production of the acid and the gas. Then the development of gas in the tube is presumptive evidence for the of the presence of coliform bacteria in the sample. First, presumptive test. This test is carried out to get information of the number of microorganisms present by means of MPN, capital MPN. It means the most probable number. The number of tubes in each group that shows gas formation in the incubation period, it gives MPN. Then confirm coliform test. This test is carried out to confirm the presence of coliform in water sample, showing the positive or the doubtful presumptive test. The samples from the positive presumptive lactose broth tube are streaked on a selective differential media, then eosin methylene blue. This medium is selective because of the presence of dimethylene blue, which inhibit the growth of gram positive bacteria and allowing the growth of gram negative bacteria. This eosin methylene blue, that is capital AMS, is differential in nature in that lactose fermenting bacteria gives colored colonies. That is a positive confirmed test due to the formation of a complex. In the presence of acid environment, AMS form a complex that precipitate out on the coliform colonies and producing the dark centers and the green metallic sheen. In this non-lactose fermenter, it produces what? That is colorless colonies which are produced on the eosin methylene blue agar medium. Then third, that is completed test. This test is one of the confirmatory tests for the presence of E. coli in the water. Then in this test, the lactose positive colonies from EMS agar, these are isolated and inoculated into a lactose broth tube, and this streaked onto this nutrient agar plate to form a gram staining. If there is production of what? If there is production of acid or the gas in the inoculated lactose broth, and there are rod shaped bacteria showing a gram negative reaction. This confirms the presence of E. coli in the water sample and indicative of positive completed test. Volume of water required in group 1, 2, 3, that is in group 10 ml. Here we can see the quantity of water required for each test. Then in set first and second, the water sample can be used directly. There is no need of dilution. Then but in set 3, instead of taking 0.0 ml of water sample in each 5 test tube, it is required to prepare 1:10 dilution by the use of phosphate dilution water, and then take 1 ml from this, and which will be equivalent to 0.1 ml of the sample. Then we will see the procedure for presumptive test. The culture media, which culture media is used for this? That is McConkey's broth medium is used for presumptive test. It is available in the market with the composition. First, that is peptic digest of animal tissue, that is it is 20 gram, then lactose 10 gram, bile salt 5 gram, sodium chloride 5 gram, then neutral lead, it is 0.75 gram. For this, first take water sample from a sewage plant and use for detection of coliform. Then make the 3 groups in test tube rack containing 3 test this test tube in each group. Then second step, add 10 ml of double strength lactose broth to the first group, that is in 3 test tubes. Then add 10 ml of single strength lactose broth to remaining other 2 groups, that is each group having 3 test tube. It means it is added into the 6th test tube. Then transfer it is aseptically 10 ml of water sample in first group of double strength lactose broth. Then transfer aseptically 0.1 ml of second group of single strength broth. Next, transfer the 1 ml of second group of single strength broth. Then incubate the all samples from 3 groups at 37 degrees Celsius for 48 hours. Then if after incubation, if there is no gas and the acid production after 48 hours, it shows negative presumptive test. If there is no formation of or no production of gas and acid after incubation, it shows the negative presumptive test, that is absence of coliform. Then if the gas is produced after incubation, then calculate the MP, and that is most probable number, and return the positive presumptive test and subject to the confirmed test. Next, that is confirmatory test. What is composition of this test? Which media is used for this test? That is brilliant green lactose bile broth fermentation tubes are used for the confirmed phase. For peptone 10 gram, then lactose 10 gram, bile salt 20 gram, brilliant green 0.013 gram, then distilled water 1 liter. First, inoculate EMS agar plate with positive 24 hours lactose broth culture with the sterile inoculating loop. Then incubate the plate for 24 to 48 hours at 37 degrees Celsius. Then absence of E. coli is negative test, indicating what? That is the water is not contaminated with fecal waste. Then if the presence of E. coli is positive, confirm test, and by use of E. coli culture perform a completed coliform test. Then inoculate 1 lactose broth and 1 nutrient agar slant from the isolated E. coli colony obtained from the EMS agar plate. Then incubate inoculated lactose broth tubes and the nutrient agar slant at the temperature of that is 37 degrees Celsius for 24 hours and perform the gram staining by the use of nutrient agar slant. This chart shows the different ambient number or the ambient index number per 100 ml. Then observation result. Examine the all lactose fermentation tubes for the production of acid and the gas after incubation and note down the result in observation. Then calculate the ambient number of given sample by the use of these values, and these ambient values of the water sample containing approximately 210 cell of the bacteria in 100 ml of sample. So what is conclusion for this? The given sample of water contain coliform and hence the water sample it is non-potable. Then question based on this practical. First, that is what is inoculation. Then second, which media is used for confirmatory coliform test. Then third, which one is primary indicator of presence of coliform bacteria. Then next, which type of bacteria are not able to grow in presence of methylene blue. And last, that is what is presumptive test.